Welcome to the Project Terra Nova. Today we're exploring why something as ordinary as dirt could be priceless. By shifting our perspective, we begin to recognize the true value of soil and the living system within it. Through this project, students learn the scientific method, including variable control, while discovering that soil is a living system essential to all life. By connecting quality in science education with sustainable agriculture and life on land, we help them see that understanding and protecting soil is key to a truly sustainable future. What comes to mind when you see a handful of dirt? How much is it worth, compared to gold? Most of us live where fertile soil is plentiful, and in that abundance, we easily forget its true value. Now imagine a place where things are very different: a barren desert, the icy expanse of Antarctica, the rocky heights of the Alps, or even another planet. Storytelling is a powerful tool. It shifts our perspective, ignites imagination, and sparks curiosity. What we often take for granted on Earth, like fertile soil, becomes incredibly precious when we consider life on Mars. Through stories, we can truly appreciate the value of our natural world and inspire new ways of thinking. So, let's imagine a human mission to Mars. What would we need to survive? Air, water, food, shelter, and even social interaction are fundamental. Thinking about these basic needs helps us understand the complex systems required for long-term space habitation, making the seemingly impossible feel a little more tangible and exciting. On Earth, we have a delicate circle of life: producers, consumers, and decomposers all working together. Could we replicate this on Mars? A closed terrarium here on Earth offers a miniature model, demonstrating how these interconnected systems function and how crucial each component is for sustaining life. A hermetosphere is essentially a closed mini-ecosystem in a jar, in which air, water, sunlight, and substrate work together to make life possible, driven by photosynthesis, the vital process behind all living things. By studying these self-contained models, we gain invaluable insights into creating sustainable environments, whether in a classroom or potentially on another planet. The core goal of the project is for students to learn the scientific method by designing and planning their own experiments, applying variable control, and discovering how real science is done. This involves asking a clear research question, forming a testable hypothesis, carefully conducting the experiment with controlled variables, and then drawing conclusions. It's about learning real scientific skills, not just memorizing facts. A fair experiment relies on understanding variables. The independent variable is the one thing you change. The dependent variable is what you measure to see the effect. And controlled variables are everything else you keep exactly the same. This ensures your results are accurate and reliable. A control group is your baseline, the normal setup without any changes. It shows what happens naturally, allowing you to compare your experimental results. This comparison is crucial for proving whether your independent variable truly made a difference, making your experiment fair and reliable. When conducting an experiment, we carefully measure our dependent variables. For plant growth, this could include plant height, the number or size of leaves, their color, the germination rate, or even how long it takes for a plant to wilt. Comparing these measurements to our control group reveals important insights. Consider an experiment where we grow plants in different types of soil or growing media. The students will find that some substrates support much healthier growth than others. This demonstrates how a single variable, like the composition of the soil, can have a profound impact on the outcome. Another compelling experiment involves comparing plant growth in light versus complete darkness. The stark difference in results clearly illustrates the critical role of sunlight in photosynthesis and overall plant health. It's a powerful lesson in the fundamental requirements for life. Our intent is to foster curiosity and exploration, guiding students to make their own discoveries. Teachers facilitate the method, not dictate the outcome. Mentoring the scientific approach is the real goal. The results are secondary. What matters is that students learn how to think, not what to think. When we let children play with science, they practice authentic inquiry. They don't just learn facts, they learn how discovery works. We've successfully demonstrated a proof of concept by growing cress in hermetospheres. This shows that even simple closed systems can support plant life, offering a tangible example of how we can begin to cultivate food in challenging environments, like those we might encounter on Mars. The students take it on from there. What about water quality for irrigation? Can we create artificial soil? Even a rather silly question is worth following up. Can plants grow when watered with soft drinks? While you as a biology teacher already know the answer, let the students figure it out on their own. So again, let's play with science, because it works. We are not done yet with our Mars mission. Decomposers are the unsung heroes of any ecosystem. Organisms like earthworms, fungi, and insects break down organic matter, recycling nutrients back into the soil. So we observe them both in nature and examine their behavior in our classroom. Understanding their role is vital for creating sustainable life on land and promoting responsible consumption and production, especially in closed Martian habitats. Imagine being able to hear the hidden life underground. Listening to living soil allows us to explore the mysterious world beneath our feet. This innovative tool makes the sounds of soil life audible, transforming observation into a multi-sensory discovery. It helps us recognize healthy living soil, whether for educational purposes with Google Teachable Machine or advanced scientific analysis. The good news is that teaching children the scientific method and variable control has a lasting positive impact on their STEM learning. When we teach children to plan and conduct experiments using variable control, we give them more than just a science skill. We give them a way of thinking. Dr. Ralf Schumacher's studies at ETH Zurich show that students who learn through structured experimentation early on develop long-term scientific literacy. They don't just remember facts, they understand how to question, test, and reason. This approach builds critical thinking, precision, and understanding of cause and effect. Abilities that extend far beyond STEM. The goal of our Project Terra Nova is simple, yet powerful: to encourage curiosity, engage children in shaping a sustainable future, and empower them to truly understand how science works. Because when they see that science works, and science matters, they realize that knowledge is the most powerful tool we have. By fostering a love for discovery and critical thinking, we equip our future generations to tackle the challenges of our world, and maybe even beyond.